We're going to talk now about novel drug delivery systems on the horizon. I'm not going to talk about long-acting cabotegravir and rilpivirine, as I would consider those approved drugs no longer part of the horizon, but we'll start with novel routes of delivery for agents that might be used in PrEP and PEP. So first, we'll talk about subcutaneous antiretrovirals, and there is one of these, lenacapavir. It is the novel HIV capsid protein inhibitor being developed by Gilead in both an oral and subcutaneous long-acting formulation platform. These are data from a phase 1 pharmacokinetic study in HIV seronegative volunteers showing the pharmacokinetic profile of lenacapavir over a year after a single subcutaneous administration of doses ranging from 300 to 900 milligrams. And what these results demonstrate is that a single 900 milligram lenacapavir dose given either as 3 separate 1 milliliter subcutaneous injections or as 2 1.5 milliliter subcutaneous injections produces plasma concentrations that remain at or above the target, which is greater than or equal to 6-fold higher than the in vitro inhibitory quotient for at least 6 months. So this suggests the possibility of a lenacapavir dosing regimen that could be used in PrEP studies, for example, with a single dose of 900 milligrams given twice yearly. Now we'll talk next about intravenous antiretrovirals, and the most active intravenous ARVs that might be used for PrEP or PEP are broadly neutralizing monoclonal antibodies. Obviously, there are several of these in clinical development. They're circled on this figure that was published in Nature in 2014. And one of the great things about monoclonal antibodies is that a single 2 amino acid modification of the Fc binding domain of the monoclonal antibody can greatly extend its plasma pharmacokinetic profile. And so here is a figure from a study published in 2019 by investigators from the Vaccine Research Center at the NIH campus in Bethesda, looking at the comparative pharmacokinetic profile of a single intravenous dose of VRC01, the unmodified modified broadly neutralizing monoclonal antibody, and compared to the modified VRC01, the VRC01LS modification in the green, showing the increase in plasma concentrations and the reduction in clearance over time, such that a single administration of VRC01LS could produce concentrations at or above the target for inhibition of HIV replication, which is 10 micrograms per mil for at least 6 months. Now, a similar monoclonal antibody in clinical development, VRC07 in the blue, also with the LS modification, has a slightly more rapid clearance and a different pharmacokinetic profile, but this is a more potent monoclonal antibody, suggesting that it too could be given once every 6 months. Now, VRC01, the unmodified monoclonal antibody, has been used in HIV prevention studies, specifically the AMP studies that were published in the New England Journal of Medicine earlier this year. And the takeaway point here is that if the virus circulating in the community was not highly sensitive to VRC01, the monoclonal antibody infusions did not reduce the incidence of HIV seroconversion. But in places where the circulating HIV virus was sensitive to VRC01, that is having a neutralization level in vitro of less than 1 microgram per mil, there was about a 70% reduction in HIV incidence with this monoclonal antibody. And so this provides some promise about the possibility of using broadly neutralizing antibodies for PrEP and PEP, although probably this will require a combination of at least 2 and possibly 3 monoclonal antibodies. Now we're going to switch from intravenous antiretrovirals to implantable antiretrovirals. And so far, there is one implantable antiretroviral in clinical development, and it is Islatrovir, previously known as MK8591 or EFDA. This is a first-in-class nucleoside reverse transcriptase translocation inhibitor, or NRTTI, and unlike previously developed NRTIs, this drug has a 3' hydroxyl in the ribose ring, allowing it to be incorporated into the HIV RNA-DNA duplex. But when that happens, it causes chain termination and prevents translocation of the HIV transcription complex, therefore giving it a unique mechanism of action and a highly potent drug that does not share resistance with most other NRTIs. Now, Islatrovir also has a very favorable pharmacokinetic profile, and this figure shows the pharmacokinetic profile of Islatrovir implants at 3 different doses used in HIV seronegative volunteers in studies that were presented in March of this year at the CROI conference. And what you can see is that all 3 of these implants produced concentrations above the threshold for HIV inhibition, which is 0.05 picograms per million cells throughout the 12 weeks when the Islatrovir implants remained in place. And once the Islatrovir implants were removed at the end of 12 weeks, the concentrations of the drug in the peripheral blood mononuclear cells diminished rapidly, consistent with what you would expect to see when this drug is given orally. So keep in mind, this is the active metabolite of this nucleoside. This is the Islatrovir triphosphate inside peripheral blood mononuclear cells in these HIV seronegative volunteers. And the drug, when it's given orally, has a half-life of over 100 hours for the triphosphate, allowing the drug to be used in this particular way. Now, the data from that study were used to extrapolate what would happen to concentrations of the Islatrovir triphosphate over a period of up to a year. And this modeling study takes the actual data, which is shown with the closed circles and the range bars, and extrapolates those data out to 52 weeks, showing that a single Islatrovir implant, at least the 56 milligram implant, would be expected to produce HIV inhibitory concentrations of Islatrovir triphosphate in a population for at least a year with a single implant. So this is really a potential game changer for PrEP. It's important to point out that in these studies, the Islatrovir implant has been very well tolerated with only some minor local reactions in the human volunteers. Now we'll next move to bioerodible implants. So unlike the Islatrovir implant, which would have to be surgically inserted and surgically removed, a bioerodible implant could be injected. And as shown in this example from Eric Apple at Stanford, once the implant precursor is injected, it forms a hydrogel through a polymerization process. And that hydrogel sits under the skin and delivers drug slowly as the gel absorbs water and expands. And in fact, gels like this have been used clinically to release drug over a period of several months. And here's an example from Eric Apple looking at the ability of a hydrogel like this to deliver a monoclonal antibody. For example, a broadly neutralizing HIV antibody could be delivered in this way. And the black squares show the antibody injected directly. The blue circles show the antibody given through the hydrogel in the same dose, functionally extending its delivery half-life from 4 days to 20 days. So the nice thing about a bioerodible implant is that it degrades on its own slowly over a period of weeks to months and would not have to be removed surgically at the end of its usable life for drug delivery. So this is another potentially promising delivery format on the horizon for PrEP and PEP. I want to talk next about long-acting oral antiretrovirals, and the oral antiretroviral that is in most advanced clinical development for prevention is Islatrovir. So here again is a pharmacokinetic plot showing the concentrations of intracellular Islatrovir triphosphate in peripheral blood mononuclear cells when this drug is given at a dose of 60 milligrams once monthly or 120 milligrams once monthly, showing that the actual data here in the blue indicate that with either dose, the Islatrovir triphosphate remains above the target threshold of 0.05 picomoles per 10 to the 6 peripheral blood mononuclear cells for 4 weeks after a single dose. And so this particular approach to HIV prevention is already in phase 2 clinical trials in individuals at risk of acquiring HIV. So again, a very exciting new format for PrEP or PEP that is a long-acting oral drug. We'll talk next about topical delivery of antiretrovirals. And of course, there is one topical antiretroviral that has already been through phase 3 clinical trials and has received an approvable, favorable review from the European Medicines Agency, and that is the dapivirine vaginal ring. Now, it's important to note that in clinical trials, the dapivirine ring was less effective than other prevention strategies. And that's largely because the device is removable. The individual who has the dapivirine ring can remove it and replace it at any time, and it's therefore susceptible to non-adherence. In fact, removal of the ring was associated with reduced efficacy in clinical trials. Now, the nice thing about a dapivirine ring is that it can be developed as a so-called combination technology. That is, in addition to the dapivirine, you can put a potent hormonal contraceptive in the ring and provide the product as both a combination hormonal contraception and HIV prevention. And it's hoped that this strategy will improve adherence and also improve the attractiveness of this approach to PrEP in low and middle income countries. It's important to point out that the development of dapivirine was unconventional in the sense that it was not developed by a standard for-profit pharmaceutical company. Instead, the dapivirine ring was developed by a not-for-profit foundation. And so it is being developed and implemented clinically in a not-for-profit setting. It's also important to note that the dapivirine ring is regulated as both a drug and device, which provides a slight complication for introducing it into generic marketplaces. Now, I want to finish by talking about transdermal antiretrovirals. And specifically, I want to talk about microarray patches, which are applied cutaneously. Now, these microarray patches have possible application to HIV prevention. And the reason for that is that a microarray patch or a microneedle array can be formulated with nanoparticles. And once that patch is applied to the skin, those nanoparticles are deposited in the subcutaneous tissue and can dissolve slowly, releasing drug over a period of weeks to months. And so here's an actual example of, in this case, a cabotegravir microarray patch releasing drug over a period of 28 days. In this case, in laboratory animals, these are microarray patch studies conducted in rats, in which the patch is applied at day 0 and then removed, but the nanoparticle depot formed by the patch releases drug over a period of 28 days. And so in the red circles and the green boxes, you can see the actual cabotegravir plasma concentrations achieved by microarray patches of 2 different kinds in a rat. Now, it's important to point out that although this is very promising and suggests we might be able to deliver drugs like cabotegravir transdermally, if you give the same drug, cabotegravir, as the long-acting intramuscular injection in rats, you achieve higher concentrations for a longer period of time. And so the blue triangles represent cabotegravir as the intramuscular injection at a dose of 2.5 milligrams, whereas the 2 microarray patches required a dose of cabotegravir nearly 5 times as high, but achieved concentrations that were lower. And so I think we still have work to do. We still have things we need to figure out about using microarray patches for drug delivery for prevention before we can present them as being equivalent to or better than other forms of drug delivery. Although I think this is a very promising technology because it can be self-administered and because it is a relatively benign way to deliver drug. So what will the future hold? I think the technology for long-acting drug delivery is evolving very rapidly. There are a lot of exciting things in the pipeline that I think will be impacting how we prevent HIV. And I think it's important to pay attention to how things are moving and which technologies are showing the greatest promise for HIV PrEP and PEP. And so I'm going to stop there. I want to acknowledge those who shared with me the slides that I have shown to you today. I also want to acknowledge my funding sources and the long-acting extended release antiretroviral resource program, LEAP. If you are interested in learning more and keeping up with the development of long-acting PrEP agents, I will refer you to our website, longactinghiv.org, which is a wealth of information about what's in the pipeline currently and what will be coming into the pipeline down the road. Thank you, and I'd be happy to answer your questions.